Good morning, everybody. My name is Igor Grigoriev. I lead fungal genomics program at the Joint Genome Institute. And it's my pleasure to welcome you all for the JGI engagement webinar series. Today, we're going to talk about MycoCosm, fungal genomics resource. So the plan for today is we'll start with a short introduction of JGI for those of you who never worked with JGI. I'll talk a little bit about the latest calls for proposals, and most of the time we'll devote to MycoCosm. We'll walk through different tools, different types of data that are available there. And so this will be a three-part video tutorial. During which I would encourage everybody to ask questions. At the bottom of your Zoom screens, you have a button Q&A. Please put your question in the box and we'll answer it either in our Q&A session or write an answer directly to you. So with that, let me introduce with that, let me say a couple of words on JGI. JGI is a Department of Energy user facility. Sorry, I'm trying to share my screen. We moved to Berkeley in December of last year. So on the right side of this slide, you see a beautiful San Francisco Bay and city of Berkeley. And on the top of the hill, it's our new building. On the left side, you see our team of 280 people, half of us in front of this new building. So half of us working in the lab producing genomics and multi-omics data, another half is processing this data and communicating with our users. So we enable large-scale multi-omics studies of plants, fungi, algae, microbial communities relevant to DOE goal of addressing energy and environmental challenges. As a Department of Energy user facility, we provide users around the world with access to high-throughput capabilities, including DNA and RNA sequencing, synthesis, metabolomics, and data analysis through community science program. So on this slide, you see various capabilities that we offer to our users. On the left side of the slide, you see our production sequencing platforms, Illumina, NovaSeq, that produce huge amounts of short reads, PacBioSQL 2 that produce much larger, longer reads. On the right side, there are examples of advanced technologies, advanced genomic technologies listed in the middle, such as DAPSeq, which allows you to detect transcription factor binding sites in vitro. Bisulfite sequencing that allows to detect DNA methylation, and ATAC-seq to detect accessible DNA regions. We also synthesize genes, pathways, and integrate them into engineered hosts. For analysis of the data that JGI produce, we partner with NERSC Supercomputer Center to run our analytical pipelines on supercomputers. All these capabilities in various combinations are available to our users via our community science program. So this is community science program is the main gateway to JGI. So there are 4 types of calls for proposals. So 2 of them are open right now, the annual large-scale call and FICO's call, joint call with JGI and AMSL. So annual call is the largest. So through this call, we offer up to 3 terabytes of sequence data. So it could be a combination of reference genomes, resequencing for all kinds of organisms, bacteria, archaea, fungi, algae, plants. We offer various flavors of transcriptomics, epigenomics, metagenomics, metatranscriptomics, DNA synthesis, and metabolomics. So the joint call with Environmental Molecular Science Lab allows users to access not only the capability I listed on JGI side, but also those available through AMSL. So various types of mass spectrometry, proteomics, imaging, and a lot of different expansive tools that they have. So they have a long catalog of tools that they offer. So both of these calls are open right now. So the proposal starts with letter of intent that are due in March. And so that's open a door for actual proposals, which are due a couple months later. And so in addition, we have twice a year, we have calls for new investigator for users who never worked with JGI in capacity of principal investigators. And functional genomics call, which focuses on combination of synthesis, DNA synthesis, metabolomics, and transcriptomics. So those two calls, the next deadline would be in July and September. In addition to community science program, we also work with industry and started JGI Industry Engagement Program or sponsored research, where we can work on partners' proprietary data based on full cost recovery model. So the next slide illustrates our evolution of fungal genomics at JGI. So the first fungal genome of white-rot fungus phanerochetic rhizosporium was sequenced and published in 2004. After that, there were many first of its kind. One example is the first mycorrhizal genome was published around this time, 15 years ago. Our largest project is 1000 fungal genome project, which aims at providing at least one reference genome for every family of fungi to build references for exploration of metagenomics data, to build a catalog of genes, enzymes and pathways for biorefineries, and to study evolution of fungi. All this data produced through various proposals processed. So for reference genome, we assemble, annotate, and integrate them into comparative system called MycoCosm. So MycoCosm currently has over 1,800 fungal genomes publicly available to users, many multi-omics data sets, all equipped with various tools for online web-based exploration, comparative genomics, genome-centric analysis, and various exploration of multi-omics data as well as community annotation tools. With this, let me move to the next step and start exploration of these tools in greater details. Let me introduce Steven Mondo, who led the development of this video tutorial. Please take it away.
Okay, thank you, Igor. And hi, everyone, and welcome to the tutorial. So I'll be presenting the first video in our three-part series covering the various omics capabilities available within our web platform, MycoCosm. So these tutorials are pre-recorded in case we experience any technical issues, so we can just keep the ball rolling. So please feel free to ask any questions that you have as we go. As Igor mentioned, please put them in the Q&A box, the Zoom Q&A box. And we'll have some time to answer those questions in between each segment, maybe one or two questions, as well as at the end of the tutorial. So let's get started. Let me share my screen. Hello, and welcome to the MycoCosm tutorial. My name is Steven Mondo, and I am a data scientist on the fungal and algal genomics team at the Joint Genome Institute. In a series of video tutorials, our team will be discussing the various omics capabilities and data available on our web platform, MycoCosm. In the first part, I will be presenting an overview of the JGI sequencing and annotation pipeline, followed by a description of the various tools we have available in MycoCosm for navigating the data. So let's get started. So MycoCosm is intentionally designed to facilitate comparative genomics. At the JGI, we perform genome sequencing, assembly, and annotation at scale to support our user community. My team specializes in genome annotation, which involves both gene modeling and functional annotation of those genes. For gene modeling, we first mask the genome to separate out transposable elements. We then align various sources of information, such as RNA-seq data and homologous proteins. After this, we employ a variety of different tools, including ab initio or de novo predictors, as well as homology and RNA-seq-based methods to predict genes. Once we have all of those genes, we functionally annotate them and use this information alongside details on gene structure to filter genes. Our aim is to identify the best model at each locus, which is then added to what we call the filtered model set. This filtered set is then used as a foundation for our gene catalog, a flexible track that can be improved by users if necessary through manual curation. All of the data we generate are first pre-released for a short period of time to collaborators for assessment and improvement, then made publicly available for the entire research community through our web platform, MycoCosm. So as you can imagine, the potential to conduct comparative genomic studies is greatly improved by the incorporation of additional genomes. One major advantage of MycoCosm is that it brings together a massive amount of data on fungal genomics. Much of these data were generated by us. However, we also actively import data sets published by the fungal community. In the past, these were imported as is, but since these come from many sources that take a variety of different approaches towards annotation, we are beginning to apply our pipeline's filtering capabilities to these external model sets to make them more comparable to the ones that we generate internally. With or without filtering, we always preserve all of the original external annotations in our portal. You will see soon how you can distinguish between these two different types of data in our portal using our navigation tools. So let's head over to MycoCosm to begin our exploration. So at MycoCosm, we have developed a variety of different ways to connect you to the data you are interested in exploring further. But first, let's zoom our browser in a little bit so we could see some more details. So before we get into these navigation tools, on this homepage, you will also see we have a sidebar which contains several important links. If you are interested in submitting a proposal, CSP calls and details can be seen by clicking this link here. We also have video tutorials available from the pull-down menu at the top of the page. Also available from the sidebar, we have a nominations page, which allows you to propose an organism for sequencing as part of the 1000 Fungal Genome Project. So this project has the goal of sequencing at least 2 representative genomes per known family of fungi. So if you have an organism from an underrepresented family that you want to sequence, you can nominate it here. Families are color-coded on this page to indicate which lineages we need more representatives from. If approved, we will sequence, assemble, and annotate the genome free of charge, as well as assist you with comparative genomic analysis for publication if necessary. So if you are ever lost within the MycoCosm platform, you can always click on the JGI MycoCosm logo at the top left-hand side of the screen, which will bring you back to the homepage. So on the sidebar, you will also see that we also have announcements of upcoming conferences and events, as well as some of the most recently released fungal genomes available within MycoCosm. So when it comes to navigating MycoCosm to identify data, your eye, of course, is immediately drawn to this cartoon phylogeny in the center, which we call the Genome Navigator. However, we also have a very useful search function immediately above it, which can help connect you to organisms you are interested in. We could type something in here to search directly, but let's click on the Search MycoCosm button over here so we could see some more details. When you click on this search button, you are immediately and by default brought to a list of all genomes we have available in MycoCosm. You can then pare this list down using the search functionality at the top. You can, for example, request to only look at genomes that were generated at the JGI or these external genomes I mentioned earlier. You can also look and request genomes that are only publicly available or published only. And the distinction between these two different types can be important because we have different data use policies for each type, which can be read in greater detail on the download pages. The last pull down here provides a list of group pages that you can then browse And if you select one of these, it will zoom in on that group of interest. If you want even more details on that group, you can click the Go to Group button on the right. However, if you already know the organism or set of organisms you are interested in, the most useful feature of this page is the keyword search. Here, you can search directly for the species you're interested in. For example, let's look for Neurospora crassa. You can either press Enter or click the Search MycoCosm button to see your results. So here you see that we have 4 different strains of Neurospora crassa available within MycoCosm, 2 of which are published, shown in green with associated references, and 2 of which are publicly available but not yet published. We can also search this page using ncbi taxonomy. Say you want to identify some close relatives to Neurospora for comparative genomic analysis. We can search for all of the genomic data available for any organism within the Sorderiaceae family, of which Neurospora is a part of, and hit Search MycoCosm, which reveals that we now have 11 genomes available for analysis within this family. So this taxonomy search functionality is based on the NCBI taxonomy. So if you are so inclined, you could even use their taxonomy IDs to filter our data. For example, let's zoom out one more step to the Sorderiales order, which corresponds to this NCBI taxonomy ID, and then search MycoCosm for that ID. You can now see that we see a list of 49 different Sorderiales genomes available for further analysis. So another neat feature of this tool I wanted to highlight was that it has the ability to search by publication. So our published genomic data are linked to papers using PubMed IDs. So if you read an interesting paper that published some new JGI omics data, you can search for that ID in our system and retrieve all of the genomes that were included in that study. For example, let's search for this publication. Now you could see all of these genomes that were part of that paper that were released as part of that paper. Also, if you're fond of a particular author's work, you can search by their name and see all of the published genomes that they're associated with within MycoCosm. So the search functionality is a great tool if you have a priori knowledge of where to look. However, if you don't have an organism, a particular organism in mind, and you want to visually explore the available data, we have 2 options for you. The first of which is the Genome Navigator on our homepage. So let's go back to our homepage by clicking on the logo at the top left-hand side of the screen. So the Navigator here is a cartoon representation showing a hierarchical taxonomic organization of the data we have in MycoCosm across fungi. You can see that some of the nodes inside the tree are named and clickable, for example, Pizizomycotina. You will also see that each leaf of the tree is similarly interactive. Clicking on any of these nodes will reveal all of the genomes that we have available at that taxonomic level and below. Note also that we have an option called Tree available here, as well as other comparative tools for analysis across the group. So we'll be talking about this tree tool very soon. So in addition to this navigator, at the top left of this image, we also have a MycoCosm group page description. So this pull down is color coded to provide a little more context on what is present within each of these groups. You can see that the vast majority of groups have no color. These are what we call phylogroups, meaning that the group is determined based off of phylogeny. We also have what are called ecogroups, which are curated groups of organisms that share a common feature independent of phylogeny. For example, brown rot fungi or ectomycorrhizal fungi. These ecogroups are highlighted in light blue. If you are interested in a particular ecogroup and do not see an organism you would expect to be there, please contact us and we will see about adding it. Also in light blue, we have other non-phylogenetic sets of organisms that are grouped based on certain criteria. For example, the 1000 Fungal Genome Project group, which contains all genomes that are part of this project. To explore a group, you can select it from the group pull down here, or you can click on that node in the navigator. and click on the top link up here. For example, let's take a look at the largest group, the fungi. So when we access a group page, we land on that group's information page over here. Other sections of the tutorial will cover these other tabs that are available for group level analysis. But this does bring us to our third way that we can connect to the data within MycoCosm and navigate it, which is this interactive tree. So the tree tool provides a searchable and interactive phylogenetic tree for exploring the evolutionary relationships between members of the group. Each phylogroup is a maximum likelihood phylogeny based off orthologous proteins determined through MCL clustering. This tree is comprised of nodes and leaves connected by branches. So each node is a bifurcation of a branch, and the color of a node indicates the support for that bifurcation. For example, the node connecting the Aspergillus and Penicillium clades is green, meaning that it is fully supported. When mousing over branches of the tree, you will also see a green box appear, which delineates the clade that will be accessed when you left-click your mouse. So as you can see here, there can be many genomes within a tree. To find one organism that you are particularly interested in, you can then zoom in on that lineage by searching with our keyword search function. So to keep consistent with our example from before, let's look for neurospora crassa and then hit this filter button or press enter. And now you can see that the tree has collapsed a bunch of different nodes and zoomed in particularly on the group that contains neurospora. So you could see also that we have grayed out all of the groups that do not fit the criteria that we just searched. So if you want to see a more normal view again, you can click this clear button to go back to more normal visibility. You can also customize your view of the tree by expanding and collapsing individual clades upon click. So for example, let's collapse the Sorteriomycetes clade by clicking back here. You can also expand it again by clicking the same node. So you can also notice that when you click on a node, it invokes this info button or info box at the top right-hand side of the screen. This information box will give you details on the support for that node, some details on ncbi taxonomy associated with the node, and how many lineages underneath that node agree with the NCBI's taxonomy identification. You can also pare down the list further to look at a zoomed in view of a particular group of interest by shift clicking on a particular node of interest. So let's shift click back here and see what happens. Now you can see that we've zoomed in on this one specific group, which contains neurospora and several outgroups. You can then kind of expand this zoom back out by clicking on this up arrow, which will move up one node each time you click on the up arrow. So we have now learned several different ways to connect and navigate the data available in MycoCosm. So let's continue our exploration of the omics data available in MycoCosm through diving into an individual genome portal. You can accomplish this using any of the approaches described today, or by clicking directly on the leaf of interest on the interactive tree, say the model Ascomycete neurospora crassa. As a model for understanding both fungi and general eukaryotic biology, neurospora has been subjected to a variety of different omics-based analyses. Our next tutorial will show you the ways MycoCosm can be used to visualize all of these different types of data. Okay, so I hope that was informative. So I see we have several questions already. So let's start with this one. I'll try and get to as many as I can before we move on to the next section. So the first question is, do you accept oomycetes to be proposed for genome sequencing or only true fungi? The answer to that is, our focus at JGI is on fungi, algae, plants, and the microbial world, but we have sequenced in the past some oomycetes. And this is also a great opportunity to kind of highlight that we have a new sister portal called MycoCosm to explore algae and other early diverging eukaryotes. So let me try and share my screen. So here's just the MycoCosm homepage, but we also at the top have a link to our sibling portal MycoCosm over here. And so we do have some oomycetes represented here under the Mycota clade. So we also have a tutorial that we just gave on MycoCosm in case anyone's interested. The videos will be posted soon. Okay, so the next question was, how exactly are eco-groups built? So let's go back to MycoCosm just to be on the home page here. So eco-groups are kind of a combination of our expertise here at JGI, as well as discussions with the user community we basically generate a manually curated set of portals that were known to be associated with a certain lifestyle. So this is somewhere we're always actively improving. So if you are interested in a particular eco-group and you don't see an organism that you expect to be there, please contact us and we'll be happy to add it in. We also have the potential to create new eco-groups if someone's interested in a particular a set of organisms. Again, just please feel free to reach out to us anytime and we'll work with you to get those eco-groups put together. So the next question was, can you use NCBI taxonomy information to search your interactive tree? Thanks, yeah, this is a good question. I didn't mention it, but yes, we do have this a keyword search capability on our interactive trees, and all of the leaves and nodes of the tree have metadata associated with them, which includes NCBI taxonomy information. So we can search for that. So let's go back to the tree. Again, you can click on any node in the navigator and then click on the interactive tree link here, which will bring you to the interactive tree. And so let's zoom in on one of my favorite groups, the Mucorales, a group of early diverging fungi. So you can hit this filter button, and then you'll see we've kind of zoomed in on this specific clade of early diverging fungi using NCBI taxonomy information. So one thing to note about the interactive tree, though, is that we build this ourselves using orthologous proteins determined using MCL. And then we run a maximum likelihood phylogeny method to reconstruct the tree. So this is totally independent of NCBI taxonomy and its construction. So there can be cases where there's discrepancies between, our phylogenetic tree and NCBI. So people have actually in the past used the tree here to kind of go back to NCBI and say, hey, this taxonomy information may be incorrect. Let's look to revise it. So hopefully that's useful for you as well. All right, so I think we should probably just keep the ball rolling here. We have more time to answer questions at the end of the tutorial as well. So let me introduce Stephen Arndt, who will lead the next section. So take it away, Stephen. Thank you.
Thanks, Stephen. Hi, everybody. I'm Stephen Arndt. I'm another member of the fungal and algal group, the MycoCosm team here at JGI. And so I'll just be showing the second of our pre-recorded tutorials that'll cover the tools and capabilities of MycoCosm in an individual genome context. So as before, just feel free to put your questions into the Q&A while the video is rolling. And again, we'll have some time after this to answer one or two of them. Hello, everyone. For this tutorial, I'll be walking through an individual portal for MycoCosm. This is the portal homepage for Neurospora crassa, a filamentous fungus in the Sordarioomycetes, an important model organism for genetics. You can navigate to this page either by searching or browsing from the MycoCosm homepage, or by using the portal ID, a unique alphanumeric identifier specific to each genome portal. These portal homepages serve as primary landing pages for genome portals. They present a broad overview of the organism, including a few brief paragraphs written by collaborators about the organism itself. This text is generally accompanied by an image or two, typically showcasing important or distinguishing morphological features. If the genome has been published, the publication of record will clearly appear on this page, and other relevant citations may also be present. MycoCosm genomes are either internally sequenced at JGI or sequenced externally by our collaborators and imported from sources such as NCBI. Irrespective of genome sequencing source, all genomes in MycoCosm will have a homepage. The homepage can also contain broad information about the particular sequencing project that an internal genome belongs to, for example, a description about the particular community science proposal that the genome was sequenced under. For external genomes, the homepage can contain text about the original source of the data. For example, here's a short disclaimer indicating that the neurospora across a genome was originally sequenced by the Broad Institute and not JGI. The prominent feature of our genome portals is this navigation toolbar. This is the way to orient yourself around the various comparative analysis tools that a portal can offer. We have search, blast, and genome browser tools, which are all ways to find things within the portal. We also have various tools for comparative analysis, including multiple annotations tab, MCL clustering tab, and a synteny browser. These comparative features are featured in a separate tutorial focused on multi-genome tools. Portals also have tabs for finding more information, like the Info tab, and getting files for a genome through the Download tab. I'll also point out here that we have context-dependent help pages for most pages on the portal. So clicking on this red help link will bring up a unique help page with more detail for that particular section. There are also some portal-specific tabs here, such as the transcriptomics, mitochondrion, and K-base tabs, which I'll cover a little later. Clicking on any tab will navigate to that page, and the highlighting at the top indicates the tab currently being viewed. So the first place we can go to is the Info page. While the home pages provide general information about the organism, the Info page will present more technical detail about the sequencing and annotation. For internal genomes sequenced at JGI, there will be additional information about the project available for logged-in users with genome access. External genomes will note the source of the data and if any filtering was applied. A paragraph at the top will provide some method details about how the genome was sequenced and assembled. There are also tables that show statistics for the assembly and annotation, for example, assembly size, N50 and L50 metrics, and the number of gene models. Finally, there are sections with external links to collaborators, the publication of record again, if applicable, internal links to our JGI Philo and Eco groups, and a short funding disclaimer. The Info page is a valuable resource, and users will likely refer to it multiple times while exploring the Genome portal. For finding genes and gene models in neurospora, we can first click on the Search tab to go to the Search page. This page lets us search in a variety of ways. We can use simple keywords, like, for example, transporter or kinase, or we can use more specific identifiers, such as PFAM, KEGG, or GO IDs. We also have internal identifiers for protein and gene models. So if those are known, they can be used here as well. We can also specify which model track to search against. This encompasses the default analysis track, the gene catalog, or all predicted models. The distinction between these tracks is most apparent for internal genomes, where many models are predicted and then filtered down into the gene catalog. For external genomes, this distinction will only be relevant when filtering has been applied, but otherwise, these tracks will largely be the same. So for example, if we search for a particular PFAM ID across the default analysis track, we can get 11 results. The results are presented in this tabular format with information about the specific gene on the left. This information includes the protein and transcript IDs, as well as the scaffold location, the name of the model, and the track that it belongs to. The next columns show any gene ontology annotations, as well as KEGG, COG, PFAM, or Interpro annotations. Here, we can see our search term in the annotations column. The final column shows any manual annotations added by logged-in users with access to this genome. Another way to find genes in the portal is by using BLAST. The BLAST tab will present a page with the option to paste or upload a query sequence or sequences to search against our portal. We can select different BLAST programs to use against a number of different sequence databases generated from the portal, such as the nuclear assembly, protein set, gene model set, and organelle assembly if available. We can modify the default program settings with these options at the bottom. The other useful way to find things is through the Genome browser. Our Genome browser is based on the UCSB gbrowse infrastructure and presents a scaffold-level view of gene organization. The browser view shows where features like predicted gene models, repeats, and aligned features are positioned along each scaffold. Near the top are our common curve tracks representing GC percentage and genome alignments against related species. Below the scaffold, represented by this black bar, are the collection of feature tracks. First is the primary gene model track in red, showing the imported genes from the Broad Institute. Below that, in blue, are BLASTX results against the NCBI NR database. Finally, at the bottom, there are 3 tracks in black, which represent the results of repetitive element identification. Some genome portals, such as this one, additionally include tracks containing multi-omics data. For instance, neurospora was part of additional experiments for assessing its growth profile on 40 different carbon sources. Therefore, this portal includes curved tracks representing select RNA-seq experiments for transcriptomic profiling in pink, and ChIP-seq and DAP-seq to identify targets of transcription factors in gold and pale green, respectively. Below the gene model track is an additional proteomics mapping track in black. At the top of the browser page, there are controls for modifying the view. We can pan the location shown on the browser by different lengths by using the different position arrows. We can zoom in or out by different factors by using the buttons under zoom, and we can also zoom in directly to the nucleotide base or amino acid level. Under general controls, you can view or download the genomic sequence of the current scaffold using the DNA button. You can set the orientation of the scaffold using the flip button, and you can open the toolbar to further customize the order and visibility of the tracks. By default, feature tracks are shown in the condensed view, meaning that overlapping features will be collapsed down together in a single row. All tracks can be reordered, and many tracks can be expanded. Here, we can expand the Broad Models track shown in red by clicking the plus button near the track, or by clicking on the track itself. This allows us to see further details about the individual models and their orientation. We can also navigate directly to a protein of interest if we know the internal protein ID. For example, this protein, with ID 2009, was identified in recent work as being a target of the XLR1 transcription factor. By using the ID in the form, we can jump directly to the model where the track has been expanded, and the model is highlighted in yellow. If we click on the individual model, we can go to that model's protein page. This landing page contains information about particular gene models. We refer to it as a protein page. The top of this page provides details such as protein name, protein ID, scaffold location, best blast hit, and any functional annotations. Some of these details contain external links out to NCBI, FungiDB, and Interpro, and others contain internal links to our genome browser and annotation pages. This next short text section contains a few additional links. The View Modify Manual Annotation link will go to the transcript page, where the primary place where registered users can add their own manual annotations. The transmembrane domain link shows text and graphics from TM-HMM annotation. There are also several graphics near the middle of the page. The blue and red graphic indicates gene structure. The coding sequence is colored in red, with any UTRs colored in blue. The intron-exon gene structure is also provided, with the lengths of introns and exons provided, respectively, below and above the graphic. Clicking anywhere on this diagram will bring up the nucleotide sequence, annotated with coding regions and UTRs where available. We can go back to the protein page by using the browser. Immediately below this graphic is a scale bar showing the length of the translated protein sequence. Any annotations where available are shown after that, signal P in purple and protein annotations in gold. Clicking the signal P text next to the graphic will show details from the signal P run. Clicking the text or images of the annotations will link to external databases. Finally, the protein structure is shown in green, with black lines indicating splice sites. Clicking anywhere on this graphic will bring up the protein FASTA sequence. Below the protein structure is a graphical representation of top blast hits, with the text serving as an external link to ncbi. The tools at the very bottom of the page will allow us to filter these blast results. For this portal, we also have a transcriptomics tab. This tab is exclusively for Neurospora crassa and represents a developmental attempt to present an interactive exploration of the large-scale transcriptomics dataset of the Neurospora ENCODE project. This heat map visualization is specifically designed for this genome and this project, but if there is any interest in presenting similar work for additional genomes, please feel free to contact us. So far, we've only been looking at the nuclear genome. A new feature we can show here is an inclusion of the mitochondrial genome. Here is the Neurospora mitochondrial genome, sequenced by the Broad and imported from ncbi. A table provides overall genome and annotation statistics, and when applicable, a link out to the original data source. The visualization shows genes colored by type surrounding the black scaffold, which can be closed or not depending on the topology of the sequenced mitochondrial genome. Arranged inside the image is a line graph of %GC, extending outward from 0 to 100%, with blue denoting GC content between 0 and 25%, and green indicating between 25 and 100%. Repetitive regions are indicated by connection lines, which will be blue or orange depending on orientation. Clicking anywhere on the image provides a higher resolution version suitable for downloading. This page will be used more frequently in the future as we roll out the capability for our pipeline to automatically annotate mitochondrial genomes. Another new feature is the addition of a tab that links to KBase, the DOE Systems Biology Knowledge Base. For a handful of organisms, there are metabolic models that the scientific community has published. We partnered with KBase to import those models into the KBase platform. Clicking on this tab will take you to a KBase narrative so you can access these models along with the corresponding genome and use KBase tools for additional analysis. KBase regularly hosts training events and webinars, so feel free to reach out to us if you're interested in learning more. Hopefully this tutorial gave you a sufficient overview of the individual portal pages of MycoCosm. We covered the tools used for searching for data of a genome in an individual context, as well as presented some portal-specific analysis tools, which will be more widespread as we continue to improve MycoCosm. Thank you for listening. Thanks, everybody. I hope that was an informative tutorial. And so we have quite a few questions, and I will answer probably one of them and then try to keep the ball rolling a little bit. See, one of these questions was, yes, can I upload my own sequences? So right now we focus on JGI sequence genomes, but we also work with PIs for external projects for species that could augment or complement existing JGI CSP projects. So if you're interested in uploading your own data, please contact us. Our information is going to be available at the very end of the webinar on a slide. So we'll have all of our contact information. So please just feel free to contact us and we can work with you to get your data into the MycoCosm system. So I'm going to keep it moving a little bit and introduce our next speaker, Sajid Haridas, who will debut the next section. Thanks a lot, guys.
Hello, everybody. So as Stephen said, my name is Sajid. As we all know, there are lots of questions on the Q&A. Wonderful. We'll answer some of them like this in between two sections, but we'll definitely get to pretty much all of them at the end of this this tutorial. So let me start by playing my section of the tutorial, and then we'll take the questions on the Q&A. So let's go. Okay, so in this part of the tutorial, we will cover comparative genomics tools available on MycoCosm. They essentially come down to three tabs: synteny, clusters, and annotations. So let's look at Synteny first. In the Synteny browser, which usually takes a minute or so to load, we can compare genome assemblies at the assembly level. So we can go genome A versus genome B. Essentially, we are aligning the scaffolds to each other to see where there is a synteny between the two. So genome A here is always your genome portal, so whatever genome portal you are on. And genome B, you get a choice. Now, because Neurospora crassa is a model organism, you have lots of choices. Most organisms don't have these many, but let's take an example here. So let's look at Neurospora hispaniola. So I click Neurospora hispaniola and click submit, and you will see that things change. So the first panel here is genome panel, and then there's a chromosome panel. And if you scroll down, there's a comparison panel. There is a wonderful help page. So if you click help, it'll pull up a help page. And so you can click the Synteny help, and that'll show you, give you an overview of what the different panels are. So what's the Synteny browser about, what the chromosome panel is about, and there is text that explains how you can navigate this. But briefly, essentially, this is showing you each scaffold, and the lines are showing you where there is more synteny versus less. So the darker the line, there is increased synteny. The chromosome panel allows you to dig down into particular scaffolds that you're interested in. So if you wanted to look at a synteny on a particular scaffold, you can look at that. And then the comparison panel allows you to look at both genomes. So you can choose in genome A as well as genome B particular scaffolds that you're interested in and look at that. So rather than go through this, do look at the help page or contact us if you need more information. There is also a dot plot. So if you click the dot plot, it'll draw a picture similar to what the mummer plot output would be like. So essentially, you are looking at genome A on the x-axis and genome B on the y-axis. And lines, of course, show synteny. You can click and drag like I'm doing here. So this works like Google Maps. You can zoom in and out using the plus and minus buttons. And then there is zoom out all the way. So if you click this button here, it zooms out and shows you the global picture. So all the scaffolds of genome A versus all the scaffolds of genome B. If you find something interesting, like here you can see there is this inversion, you can use this plus button and then zoom in. Right, so you can just select that and it'll zoom into that section. So you can see that supercontext 6 of Neurospora crassa versus Neurospora hispaniola scaffold 4. You can see this inversion, and then you can go in and look at additional things on the genome browser that you might be interested in. So let's go back to the standard view by clicking Synteny again. This will bring you back to the genome portal. So this is 1:1 genome comparison. So let's look at multiple genome comparisons, which you can look at on the annotations page. When you click the down arrow, you can see that we functionally annotate the genes on many different levels. So let's look at gene ontology as an example. So when you click gene ontology, you will come up to a page that shows you the genes that are involved in particular Go processes, right? So biological processes or cellular component or molecular function. And you can use the plus and minus to essentially expand this and dig down into particular things that you might be interested in. You can also directly search using the search term or search accession. But the interesting part here is you can compare this organism, which shows up by default, with several others that are available here. So you can shift click to select several, or you can control click. So I'm just going to click the Neurospora hispaniola here and click apply. And so what this will do is this will expand this table to include both Neurospora crassa here, and now it is showing Neurospora hispaniola, and then you can compare between the two. So you can see that hispaniola has fewer Go terms associated with it, and if you click the plus, for example, you can drill down and it'll go here. I have clicked molecular function, and it'll drill down into that, and then you can further drill down and so on. Okay, so this is how you would look at Go activity. Of course, you can click on the link that you might be interested in. So for example, if you wanted to look at these 29 genes, these 17 genes, or these 46 genes, you could click at those. So for example, if I click here, those 29 genes, it'll bring me to a page that shows those particular genes with that Go annotation, right? So here are those 29 genes. It'll show me the protein. I can download the proteins or the transcripts, and there is additional details on what the Go is. So there are link outs and stuff like that. Okay, so other annotation pages look in the same vein, but it might look slightly different. For example, if I click on PFAM domains here, you will see, again, it's a tabular view, but in this case, all the comparative genomes show up by default. So if you mouse over, it'll tell you what these are. So this is our genome of interest, Neurospora crassa OR74A, and then all of these others are other Neurospora strains that we have. And so this is, again, just a table with link outs to the PFAM domain, and a table, all of which is clickable, that shows the genes with that particular PFAM annotation. So for example, if you wanted to explore the ABC transporters, you can click the 279 for all the ABC transporters in all of these genomes. Or if you were just interested in Neurospora crassa, you would click this 37, which is under the Neurospora crassa, right? So you would click here, and then it'll bring up the 37 ABC transporters in Neurospora crassa. This is the detailed view. So the first column, it gives you the protein ID. So that will link you to the protein page, which you have seen previously. The location, which will take you to the genome browser, again, which you have seen previously. And then there is statistics about how long the gene is at the nucleotide level, how big the protein is, what PFAM domains are on this particular protein. So for example, some have multiple PFAM domains, and then the actual model itself, as in the exon intron structure. The clicking on this domains tab here will show you where on that protein the domains were identified and what the e-value associated with those proteins were. So rather than go through all the other annotations, they all work similarly. So you will start with a table, and then you can dig in to particular things that you're interested in, or use the search functions that are available, right? So in all of the annotation pages, there is search function like here in the PFAM. Okay, so now you know that we have all of these annotations. The good part is you can download all of them. So if you click the download page, this is the one page where you need to have an account at JGI to download the data. So I am just going to go to a different browser where I have logged in. So here is the same Neurospora crassa page, and I have logged in here as myself. And so now when I click download, so it will give me the download page. So the first part, since this genome is public, it'll give you the reference that you can use. You click OK, and it will take you to the page where the actual files are. You can dig down using this plus, and then dig down further, or you can just click expand all here. So when I click expand all, it'll show me all the files that are available. And as you can see, we have the functional annotations of it we were just looking at. We have the KEGG, the COG, the Go, and signal P, and so on and so forth. In addition, we also have the assembly, both masked and unmasked, and the gene models in GFF3 and FASTA form. So all the download is available. I want to take a slight diversion. So this is an external genome, as we started this presentation with. If you look at an internal genome, so this is Neurospora hispaniola, which was assembled and annotated at the Joint Genome Institute. So the download page here looks slightly different. So when I click download here, the first thing it'll tell you, so this genome is not yet published. So we have a data release policy that basically says, feel free to look at the data, use it for your own research. But if you want to publish, please talk to us and talk to the PI that provided us the data. This is just good practice. I hope you will all agree. So once you click agree, it'll take you to a very similar looking download page where again you can drill down. But I'm just going to click like before, expand all to show you one major difference, which is you have the same sets of files. So you have your functional annotation, you have your assembly data, but in addition, you have this additional set of files that says all proteins, all proteins and all transcripts. As you know, the JGI uses multiple gene calling methods and then a filtering algorithm to come up with the best gene at each locus. So the all genes is essentially gene calls from each of those individual predictors. So each genome could have, 100,000 genes in this all genes, for example. So in whether you're using an internal genome or an external genome, what we think is the best set of genes for that genome remains the gene catalog. So both here in this internal genome, you have gene catalog. And if you go to the external genome, again, you have the gene catalog. So that's what you should be working with. The all genes is more if you're interested to look at how the different predictors performed in each particular case. This may not be of interest to all users, but if you are, the data is there. So I'm going back to my other browser and closing this window for the download. So now we have these annotations. Let's look at another way that we can compare genomes, which is using MCL cluster. Okay, so the MCL clusters page uses MCL clustering methods to cluster genes based on similarity. So it's an all versus all blast of all the proteins in all of these genomes. And then they are made into groups, right? MCL clusters. So here, each row represents a cluster and the columns are the various genomes in that particular clustering run. Many genomes will have multiple options for clustering runs. Neurospora crassa, being a model organism, is an outlier. So there are a lot of clustering runs. Most will have maybe a couple or 3 or 4. So you'll also find cases like this. So for example, these two have the same name. Here in this case, the later number is the more recent clustering run. So let's select that one. So you can see a few more genomes have shown up in the comparative tool. You can sort them by clicking on the header. So if you sort based on Neurospora crassa or 74A, the largest number of genes clustered in that genome show up on top. There are other ways to filter, including by the number of genes in a particular cluster, either globally or for a single genome that you might be interested in. You can use search terms. So for example, ATPase, right? So it'll show up clusters with that particular functional annotation. And then you can download this data as CSV or FASTA files using the download button. By default, everything that you see is downloaded, but you can select particular clusters for download by selecting using the checkboxes here. As usual, there is a wonderful help page. Feel free to take a look at how these clusters are determined and additional help on how to navigate this page. But each cluster has additional cluster details. So if I click a particular cluster, I'm clicking 22, it'll show all the genes in that cluster. Protein ID and genome name shows up in the first column, like we saw on the PFAMS page. Location on the scaffold, gene length, protein length, and the domains associated with it. And here in the last column, you have the same as before. We have models, the intron exon structure, domains, which you've again seen in the PFAM page, with the location of this particular associated PFAM. But this new tab shows up synteny, which essentially shows you the local location of this particular gene in this cluster in relation to other genes on that scaffold. So the central gray shows you the genes in that cluster, and then it shows you 5 streams upstream and 5 genes upstream and 5 genes downstream. And the colors represent the other genes, whether they belong to particular clusters or not. So for example, this one is cluster 22 since I selected it. But if I click, so for example, if I click this dark blue one, you can see that this is cluster 2742, and there are other members of that cluster which also light up, like that one there and that one there. So you can directly go to that cluster by clicking the link here. So that is MCL clusters page. Okay, so so now we have looked at all the comparative tools that are available. So I want to bring up one more thing, which is what if you wanted to look at a different set of comparative organisms rather than the one that's available here. So this goes back to the initial presentations where we spoke about group pages. So I'm going to go to the info page for Neurospora crassa here. And right here at the bottom, there is links to group pages. Phylogroups, groups, as you know, are linked by phylogeny. And eco-groups, which this genome is not a part of, but eco-groups are manually curated lists of groups that might share some kind of some common thread. For example, plant pathogens is a group of plant pathogens in MycoCosm. So the largest group, of course, is fungi. The smallest in this case is the genus Neurospora. So if you click Neurospora, it'll show you all the Neurosporas that we have. But let's go one step up and look at the Sodariaceae. So I'm clicking Sodariaceae, and now you see that we have Neurospora, and then we have a couple of Sodaria genomes. So this is a larger group than what we were working with. And this group portal has the same functionality as an individual portal. Of course, there's no concept of one-to-one synteny, so there is no synteny tab, but things like clusters and annotations. So the same deal remains, right? So you click now PFAM, and now you have PFAM across all of the members of that particular group. Similarly, you can click blast, and now you can blast all of the members of this particular group, whatever group you are on. In this particular case, Sodariaceae. Search also works the same way. So rather than searching one individual genome portal, you can search the entire group. The one new thing here that shows up is tree. So this is much like what you saw in the introductory tutorial section. So you have a tree of the Sodariaceae itself. So that essentially are the tools available on MycoCosm, both at the individual portal level and now these group pages. Help pages are always wonderful, and I hope you found this useful, and we are ready to take questions, and feel free to contact us whenever you need help. Thank you. So we are back. I hope you found that useful. There are several questions. There's one interesting one on download. Download has always elicited questions. So I'll try and shoot two birds with one stone. So let me share my screen here. Let me share, here we go. So, so this is, I'll go to Neurospora. So we'll go to the Neurospora Hispaniola website. So Stephen mentioned this earlier. So all you need is the portal ID, in this case, NewHe, which is the portal ID for Neurospora Hispaniola. So if you put NewHe1 and hit enter, that'll take you to the Neurospora Hispaniola site. This is also wonderful. So anytime you contact us, if you tell us NewHe1, we know exactly what you're talking about. You don't need to give us the full name. So this is the Neurospora Hispaniola. I am logged in. So when I click download, it will show me the download page. And then once I say agree, sorry, you'll see me moving around since I'm working with multiple monitors. And then as before, I'll hit expand all. And so we have several files. And so there was a question on what these files are. So promoters, we are essentially going 1 KB upstream of the gene. That's pretty much it. So there is an all protein and transcript ID map. As I said, protein IDs are unique within a portal. And so just like we have protein IDs, we have transcript IDs. So this is the mapping of the protein ID to transcript ID. They are just numbers, they are numeric. And so if you want to map a transcript to its protein, so this is the file that you would use. It's just a text file. As I mentioned, we have the all CDS and all genes. I hope this is self-explanatory. If not, type your question in again and I'll give you additional details. So for now, I'm going to skip this all because this is used by a very small subset of our users. The important part is here, gene catalog. FASTA file, CDS, of course, is the CDS, right? The KEG, GO, InterProScan, and COG are functional annotations. Signal P is where we scan these proteins for signal peptides. GFF3 is the gene catalog in GFF3 format if you wanted to use it in your own local environment to load and see where the genes are. GFF is, of course, is the older format. And the FASTA file, so the amino acids, so these are the proteins, and the nucleotides, so the CDS is in FASTA form. The assembly scaffolds, this is the masked version and the unmasked version. And as you know, where we have mitochondrial genomes available, that's also available for download. Hopefully that was good enough. There was also a question about downloading using an API. And so if you click the help button here, sorry, it opened in a new window. So here's the help button. And there is help on downloading with API. So for those who use API, click this. And if this is not clear, write to us. We will give you our contact information at the end. It should be pretty straightforward. If you have trouble, we are always here to help. Hope that helps.
Okay, thank you, Sujit, Stephen and Stephen, for brief introduction. So thank you everybody for starting asking us questions. So please don't stop, continue. So for the next 12 minutes, we'll try to answer all your questions. If that's not enough time, we'll continue after we're done. So the group of the questions that we typically receive is what makes, I spoke a little bit about CSP calls for proposals, right? And there are questions on that topic. What makes a successful proposal? How long should be a letter of intent? What is the funding rate for CSPs? Letter of intent is a short web form. So there are special sections that you need to fill in. Every proposal should have a connection to DOE mission. So that's one criteria for success, which is bioenergy, carbon cycle, and biogeochemistry. So cool science always win. And the third criteria is a user community. So whatever we do, we try to serve larger user communities. So the data that will be generated as part of these CSP proposals, if they're utilized by a large group of people, so that's always better than when it's a single lab research. So when you think about proposals, think about your collaborators, colleagues, team together, try to answer larger questions through expertise from different labs, as well as through different products that we offer within these calls. Funding level, so first, we don't necessarily fund your research directly, so we provide these resources for free. So this is different than how other funding agencies work. And we aim to deliver a complete package, not only sequence and produce synthesis, and we also do all bioinformatics analysis, and we're trying to work with our PIs and their teams to analyze the data, to process the data into biological stories, hopefully publishable biological stories. And about 25 to 30% of the proposals on average are selected every year. So it's pretty high in comparison to other funding agencies. If you have any questions, so I know the deadlines for letters of intents are close. So in a week, it's a deadline for JGI AMSL. And by the end of the month, for large annual call, but we're always open to answer your questions about CHPs. All right, so moving forward, one question was about how many genes are annotated. And this is an interesting question. This is also a segue in something in a new initiative that we're trying to develop. And I'll let Stephen Aaron to answer that.
Yes, so there is a broad range of functional annotation across the fungal kingdom. So some genomes like yeast have a lot of well-characterized genes. Other clades like the Rus have maybe up to 70% uncharacterized. And so we're actually working on a tool. Let me share my screen. So this is, yes, so this is, we're looking at conservative families of genes with unknown functions, looking across the entirety of MycoCosm. So this is kind of a tool that we're developing to present candidates of these genome clusters to the community. So what we've done is we've taken like 18 million proteins from 1,300 fungal genomes in MycoCosm, cluster them together, and present here a subset of 142 of them that are not only conserved across large phylogenetic distances, but have unknown or poorly characterized function and also supported by transcriptomics data. So here on this page, the columns start with an identifier for the cluster itself, give the number of genes and the number of unique species that cluster covers. And then some columns about the distribution of these unknown PFAM domains, how many there are, how many proteins have them, etc. We also provide some hints that might let you guess at the function. These are HMM searches against the UniProt and PDB database. We also show kind of a broad taxonomic distribution of the models in these clusters, and their average lengths and whether any of them already have existing manual curation in them. And so these are kind of open to the community for adoption, as it were. And so you can go to one of these clusters, click on the number, there's this add note, and you click on that and you can indicate your either progress if you're kind of already working on this, or your intent to move forward and do manual or experimental curation or other characterization of these. And so this is, if you're interested in participating in this this type of initiative, reach out to us and we can provide this tool in this page to you for your work.
Okay, great. Thank you, Stephen. So this is a work in progress. This is really a pilot that we developed. So before prime time, if there are any volunteers to look at some of these gene families and characterize them, we can share the link. So it's not connected to MycoCosm yet. We're still in testing mode, but once some volunteers check this tool, we're more than happy to make another page in MycoCosm and open it to broader community. So this is kind of, so the idea is that you use direct biochemistry characterization for some of these genes, and since they're conserved, you can propagate this annotation across the entire tree of life. So they conserve, they're important, nothing is known about them. So I think this could be very rewarding. So this is also an opportunity to think about multi-omics capabilities available within those calls and apply them to characterize some of that, maybe in combination with some mutagenesis analysis, there are approaches like TNSEQ or insertional mutagenesis. We can do sequencing part to do barcoding, for example. There are many CRISPR-Cas9 systems developed in various organisms. If you have mutants, we can do multi-omics characterization of all of those mutants as well. So think about those opportunities. I just shared our emails, check with us, and we will share the link for this page. We're looking for feedback and also for people who would like to do this. Okay, continuing with these questions. So I guess one of the questions was, how do I find my favorite gene in your portal? So maybe that's a question to Stephen Monda.
Sure, yeah. So the question was asking about if there's a possibility to search genes or proteins by their commonly used names in publications, such as ERG1 or something like that. This is a great question. So we don't yet have capabilities to scrape publications for common names and load them into our system, but we do have the capability to have, users can manually curate genes and add names to particular portals. So one example portal like that is the Aspergillus flavus portal. So let me just share my screen. This is one of our attempts to make kind of like a gold standard genome assembly and annotation. So it's a complete chromosome level assembly. And so the research community here has done a lot of work trying to pair these genes that they know have a certain function with their names, so they're searchable by the community. So here's just one example. We can go to the search page and we can look for something like PPOA, and we could search for it here, and we'll get some information that our colleague Jeff Skirker loaded in with the name of the gene, and they put some literature in here where that name came from, which will make it very useful to search. So another question that came up, which is somewhat related, was from Carlos, which was asking about when looking for a protein ID from a different strain, sorry, not a different strain, from the same strain but a different assembly. Do we have a way to look at that in the newer portal that's generated? And so this comes to we have a method that we call mapping forward, where we will try to take an older genome annotation and map it forward onto a newer assembly. And in that way, we preserve those old protein IDs from the original annotation on the new portal. So his question was specifically about Mucorsucinoloides MU402. So I've already kind of loaded that page up. So this is MU402. And I just wanted to highlight the mapping forward track that we generate here. So there's a couple ways that you can search for these models in the genome. So you can look visually on the browser, and you can look for your, compare the old models to the new ones in the gene catalog. You can also search for those models by their protein ID on the search page. That's a possibility. And then another thing that you could think about is if you have a newer model in your gene catalog that is, different from the original one, you can then search for your old protein ID here, find that location on the genome browser, and then, jump from that one to the new model. We have some other developmental efforts to try and better connect these with our search functionality. So I'll just pop right back to the Flavus portal for just a moment, where we have been doing work to try and overlap the older gene models with the newer ones so that the community can use the same old names they're familiar with to search for their genes of interest. So here I'm just going to do a basic search across the gene catalog, or actually what'll be better is to look for user annotations, where we have loaded in information on what overlapping models came from the original source, in this case, FungiDB. So then you could see the newer model, and you can connect directly to the old one as well. So hopefully that was sufficient, helpful for answering those questions.
Okay, great.
We'd like
great,
to
Stephen.
move forward.
Yeah, thank you. So another question that's probably a question to Sajid. He talked about various types of comparisons. So there are several questions that sort of in this area. So one was, what other statistics beyond genome size and gene counts we show. He demonstrated a few targeted annotations that allows you to compare numbers side by side. So the question is, are all annotations based on PFAM? First part of the question, and is it possible to download these annotations? Yeah, please go ahead.
Is it possible to download the annotation? So let's take, so I demonstrated the download page, and so all the functional annotation is available for download. As far as the annotation pages are concerned, let me share again. So within the annotations, so PFAM is primary. Transcription factors are essentially a subset of the PFAMs that are known transcription factors. So you will find that it has the same structure, PFAM. The others, peptidases, transporters, they are available for download. Secondary metabolism clusters, we have essentially developed something in-house that works similar to SMRF, if you know about it. And K-zymes, we have had a long-standing relationship with Bernard Hendershot's lab at kz.org, and they expertly curate K-zymes for us. So there is sometimes a delay between us releasing a genome and the K-zyme data becoming available, but these are all curated by Bernard's group. Sorry, Igor, what was the other part of the question? I missed it.
They're based on PFAMs. Yeah, so are they based on PFAMs and whether they're available? I think you covered.
Okay.
So,
sorry.
Yeah, we have algorithms for each type of annotation, and we keep working on those, making them more accurate and expanding the collection of gene families, types of the families that we can annotate.
And just to make this a little bit more interactive, I also wanted to tie into what Stephen said previously about finding genes that you might be interested in. So another way to think about finding genes that you might be interested in is you have a gene of interest in your genome of interest. What about homologs in other genomes, right? So for example, let's say you wanted to find a particular kinase that you're interested in. So if you know the protein ID, so in this case, protein ID 960 in Neurospora crassa, so NuCRS2, I'm going to refer to these by portal IDs. Then you can go to the MCL clusters page and find that. Now remember, protein IDs are unique in a genome, but not unique across genomes. So in order to find 960, you really want to make it specific to that genome. So I will put NuCRS2, a dot, and 960. This search strategy is there, available on the help page, by the way. So even if you don't remember it, go to the help page, and I'll press enter, and it will pull up the cluster with that particular protein in it. So then you can find the homologs in other genomes. So if you click on the cluster, here is your 960, and it'll show you what we've discussed before, right? Its location, its syntonic context, etc. I hope that helps.
Okay, yeah, and while you were answering, so one question was whether we're planning to add cytochrome P450. So yeah, it's in development right now. We have our internal procedure, so it should be added at some point. So as I said, we're working on expansion the collection of targeted annotations. So another group of questions, maybe Stephen Arndt can address them. So several of them. So is it better to use filtered than non-filtered models? You mentioned, so that was one. Second, you start talking about filtering external genomes. What's exactly does it mean? And three, how do you handle deployed organisms?
Yes, those are all fantastic questions. I would say that it is probably best to use the filtered model set. Just that is kind of the attempt to make everything comparable. And so if you're looking between organisms, that's the best guess at the unique set of functional capabilities for an organism. It's based on we have many different gene modelers that predict a a certain model for a particular locus, and then we filter we select kind of heuristically the best one. And if there's perhaps a discrepancy there, the non-filtered set is also available for you to explore to kind of try to address that. So I would say yes, the filtered is probably the best, but we also offer the rest of it for your own comparisons. For importing genomes, yes. So this filtering, it's trying to make these externally sequenced and external models much more comparable with our internal ones that are filtered by default. And so this is a recent developmental attempt. And so we're basically removing things like TEs, pseudogenes, overlapping models, alternative transcripts, alleles, and these unsupported short models. And in that way, we can make it more comparable to what we've done with our internal genomes. And of course, I think Stephen also mentioned this, but yes, we do keep the original external model set with no changes and no modifications at all. And so for external models for external genomes that are imported and filtered, we provide both of these sets in the downloads page. And for diploid genomes, yes, we do. We do those predictions through our annotation pipeline. We identify scaffolds that we think are allelic, and then we predict the models from those scaffolds, and we assign them as primary or secondary models. And so those are presented on a portal as a separate track underneath the existing filtered model set. We have a primary and secondary allele track that shows those, and then those are also available in the downloads file or in the downloads section.
Okay, thank you, Stephen. Okay, so we're running out of time. So let me thank Sajid, Stephen for bringing the tutorials live. So these tutorials will be linked to MycoCosm. This whole recording will be available via JGI webinars. I shared the link with everybody. I also shared a few links sort of if you're looking for ideas, inspiration, and like to get better understanding how to put together a proposal. There is a number of webinars, there is a number of podcasts, and we just released the JGI progress report. We'll stay and try to answer all the questions. And if you know your contact information, we'll get back to you. If you don't hear from us, please don't hesitate to email us directly. So this is our contacts. Please think about a new proposal. Check with us if it's a good idea, what kind of capability we have. If there are any questions on MycoCosm, feel free to email to us as well. So we hope that this webinar was useful, and again, it will be available, and we'll try to convert questions and answers into FAQ page. Thank you very much for staying with us. We're looking forward to see you in our future webinars.